So, chapter 2 is going to cover like the basic chemistry and physiological reactions. Our body's going to be made up of many chemicals, and the chemistry underlies all physiological reactions that happen: movement, digestion, pumping of the heart, the nervous system. So, in this chapter, we're going to look at how chemistry can be broken down into basic chemistry and biochemistry. Now, I have a video that's a good introduction to this chapter. Of course, you still need to read the chapter. Make sure you do your assignments at the end of the chapter.
I know, this is biology, but we have to start with the foundation in chemistry. If we don't focus on the microscopic first, then nothing that happens on the macroscopic level will make any sense. A strong understanding on what influences the chemical behavior of an atom will help you draw connections between molecular structure and function and make predictions for cellular processes. Every organism needs to obtain energy and exchange matter with the environment. So let's start with an introduction to water and review the most common elements and reactions for life. Then I'll recap the 4 groups of biological molecules: carbohydrates, proteins, lipids, and nucleic acids, which will make many appearances in all 8 units of AP Bio. Let's zoom in. Water might be the most amazing molecule on Earth, and it can defy gravity. How? Well, I'll tell you, but we need to sketch it out first. Good old H2O. 2 elements of hydrogen for every element of oxygen form with polar covalent bonds. Oxygen is more electronegative and has more pull on the shared electrons with hydrogen. Consequently, oxygen has a partial negative charge, while each hydrogen atom has a partial positive charge. This uneven distribution of charge results in a polar molecule of water that makes life possible. If we can recall our chemistry background, we know that opposite charges attract and like charges repel. Because water is polar, it will attract to and bond with other polar or charged molecules, even earning the title the universal solvent. In fact, almost all of water's unique properties are due to its polarity and ability to form hydrogen bonds. Because opposite charges attract, a hydrogen bond forms between a partial positive hydrogen of one water molecule and a partial negative oxygen of another water molecule. When we look at water as a collective, we'll find hydrogen bonds forming between adjacent water molecules and polar covalent bonds forming within water molecules. Hydrogen bonding is the foundation by which water molecules interact with other things. In liquid water, hydrogen bonds are continuously breaking and reforming. Boiling water has to break those hydrogen bonds in order to cause the water molecules to move more rapidly, which is why water has a high specific heat. However, when water freezes, the hydrogen bonds become more stable and the water molecules get pushed farther apart and are less dense. This is why ice floats on water. The 3 big properties of water you need to know for the exam are adhesion, cohesion, and surface tension. Water is attracted to water, called cohesion, and to other polar or charged things, called adhesion. Both cohesion and adhesion give water its remarkable ability to defy gravity. Let's look at a plant. Water moves vertically from the roots to the leaves through adhesion to the walls of plant vasculature and cohesion to other water molecules. As transpiration occurs at the leaves, there is a barrel of monkey action in which water molecules pull up on the water molecule below it. You might have also seen this gravity-defying stunt in a graduated cylinder during lab. A meniscus forms at the bottom curve of a graduated cylinder because the forces of adhesion to the glass are stronger than cohesion between water molecules. Water literally jumps up the sides. Water also has remarkable surface tension due to cohesive and adhesive forces, as seen in a droplet of water or morning dew that forms on a blade of grass. Surface tension is also why certain insects can literally walk on water. The interaction their body has with the water is weaker than the hydrogen bonds between the water molecules themselves. Organisms must exchange matter with the environment to grow, reproduce, and keep themselves organized. Quick science background review. Matter is made up of atoms, has mass, and takes up space. For example, rocks, trees, and people are made of matter, while energy is not. Atoms contain 3 subatomic particles. Protons are positively charged, found in the nucleus, and are represented as the atomic number on the periodic table. Neutrons have no charge and are also found in the nucleus. Electrons have a negative charge and are found surrounding the nucleus in rings. Because electrons are the most peripheral of the 3 subatomic particles, they will be involved in bonding with other atoms. Metabolism is the sum of all chemical reactions that take place within an organism. those that are bond-breaking or catabolic, and those that are bond-forming or anabolic. We have to do both to be biologically successful. When organisms obtain matter, they can use the raw materials to build useful molecules for biological processes. For example, organisms take in glucose as a reactant for cellular respiration and carbon dioxide for photosynthesis. What do you think of when you hear the word organic? Images might come to mind of apples grown without pesticides or grass-fed cows. But in science, organic molecules contain carbon. Because carbon has 4 valence or outer ring electrons, it forms 4 covalent bonds with other elements. These bonds can be single, double, or triple, causing molecular shapes of straight chains, branched chains, and rings. Carbon is so versatile. The 6 most common elements found in living things are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Carbon, hydrogen, and oxygen are represented in all biological molecules. Nitrogen will never be found in carbs and lipids, but is always a component of nucleic acids in their nitrogenous base, and proteins and the amino group. Even though nitrogen is the most abundant gas in the atmosphere, it will need to be taken in through food or absorbed from the soil. The final 2 elements make infrequent appearances. Phosphorus will always be in nucleic acids as a component of the backbone of DNA, and it will appear in lipids that are phospholipids. The presence of a phosphate group gives DNA a negative charge, and phospholipid heads their polarity and subsequent hydrophilic affinity. Sulfur makes its only appearance in some R groups of proteins and influences protein folding through the formation of disulfide bridges. Water is a key player in 2 of our primary chemical reactions of metabolism. Dehydration reactions are used to join monomers together and form a larger polymer. It's exactly as described: to dehydrate, to take out water. So, if you link 2 adjacent monomers, you can pull hydroxyl from one end and a hydrogen off of the other, removing H2O. The electrons left behind on those monomers are now available to covalently bond together. In contrast, hydrolysis reactions add water to break a polymer into monomers. When this occurs, we stick a hydroxyl onto one monomer and a hydrogen onto the other. The remaining 3 sections of Unit 1 cover the 4 biological macromolecules and properties, structures, and functions. Instead of jumping back and forth for each type within sections 1.4, 1.5, and 1.6, I'm going to review each macromolecule individually, beginning with proteins, then carbohydrates, lipids, and lastly, nucleic acids. Proteins are polymers formed from amino acid monomers. An amino acid is made up of one central carbon atom that forms 4 single covalent bonds. One is to hydrogen, another is to an acidic carboxyl group, and a third is to a basic amino group. The 4th covalent bond is to an R group, which is a substitute for different side chains that give each of the 20 different amino acids their unique chemical properties. These properties could be hydrophobic, hydrophilic, or even ionic. When amino acids link together, dehydration synthesis occurs between an amino group of one monomer and the carboxyl group of the other. The covalent bond formed between them is referred to as a peptide bond. And when you get many peptide bonds in a row, you formed a polypeptide. Polypeptide chains, the primary structure of proteins, are formed during the process of translation at a ribosome. In eukaryotes, the newly synthesized polypeptide is released for further processing by the rough endoplasmic reticulum and shipping by the Golgi body. Several scientists were initially convinced that proteins were the molecule of heredity, not DNA. It made sense. They have more alphabet in their chemistry than its counterpart DNA, with 20 amino acids as compared to 4 nitrogenous bases. And you can form a lot more diverse structures for a multitude of functions with proteins than you can with DNA. Proteins have 4 levels of structure as it bends and folds in upon itself. One wrong amino acid and the protein folding will be incorrect, potentially causing an error in function. Primary structure is a chain of amino acids from the N terminus to the C terminus. Secondary can either form an alpha helix or a beta pleated sheet through local folding from backbone hydrogen bonding. Tertiary structure has a specific 3D shape when alpha helices and beta sheets fold further inwards due to R group bonding interactions. The final level of structure is quaternary, which has 2 or more separate amino acid chains interacting and bonding together. The specific structure that each protein has is dependent upon the chemical properties of the R group and can be influenced by environmental factors. R group side chains that are hydrophilic or charged fold outward toward the aqueous environment, while those that are hydrophobic face the interior of the protein. Protein structure denatures when hydrogen bonds are disrupted. This shape change directly affects the protein's function and can sometimes be reversed since primary protein structure is not changed. Proteins practically do everything for the cell, and even this impressive list I'll discuss now is incomplete. They are a key component in cellular membranes with roles in transport, recognition, movement, and communication. Integral membrane proteins have hydrophobic regions that interact with phospholipid tails and hydrophilic regions that are adjacent to the heads. Some integral proteins have specific molecular chemistry internally, forming a channel. These channels may be gated and allow specific ions or small molecules to cross the membrane through facilitated diffusion or active transport. Peripheral proteins like hormones and antigens, they're more loosely attached to the membrane and are involved in cell recognition and communication. Membrane proteins can also provide anchorage for the cytoskeleton, aiding in structural support and cellular movement. And lastly, proteins also form enzymes that catalyze chemical reactions for specific substrates. For example, the enzyme sucrase has a specific active site which fits the substrate sucrose, a disaccharide. This enzyme facilitates the hydrolysis reaction of sucrose into monomers, glucose, and fructose. Carbohydrates only contain the elements carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. They are polysaccharides formed from monosaccharide monomers, which join to other monosaccharides with a specific type of covalent bond called a glycosidic linkage. These biological molecules can form long chains or ringed structures. Formed by autotrophs and disassembled by nearly all living things, carbohydrates are the primary source of chemical energy for the cell and serve as structural material. Monosaccharides like glucose and fructose, as well as disaccharides such as lactose and sucrose, are often used as fuel sources for the cell. Glucose, C6H12O6, takes the main stage in AP bio during cellular respiration in the mitochondria and photosynthesis in the chloroplast. Larger polysaccharides, such as cellulose in plant cell walls and chitin in animal exoskeletons, can be used for structural support. The polysaccharides starch and glycogen are used for long-term energy storage in plants and animals respectively. The primary contrast in types of polysaccharides is in their glycosidic linkage, branching patterns, and functions. Many carbs look like chemical twins at first sight, but organisms can tell the difference. Glucose has an identical chemical formula to the isomers fructose and galactose, but its atoms are arranged differently. Starch and cellulose are both formed from consecutive glucose monomers, but have varying linkage at the 1,4 bond. Even these small differences are enough for enzyme substrate specificity, which is why glucose is used for cellular respiration, not fructose, and animals can digest starch, but not cellulose. Lipids are a very diverse group of hydrophobic molecules that are nonpolar due to their hydrocarbon chains. They contain the elements carbon, hydrogen, and oxygen, and sometimes phosphorus when forming membranes. Unlike carbohydrates, lipids have a ratio of hydrogen to oxygen that is much greater than 2 to 1. Instead of an elemental ratio of 1 to 2 to 1 like carbs, you could say 1 to 2 to few. Additionally, lipids don't have a true monomer, as there's no structural pattern of repeating units. The different lipid varieties, fats, phospholipids, and steroids, all have unique structures for specific roles within organisms. Fats contain a glycerol and long hydrocarbon fatty acid chains. A saturated fatty acid is named for a literal elemental saturation of hydrogens. If all electrons within a hydrocarbon chain are occupied with single bonds to hydrogen, then the fatty acid tails are straight and more closely packed together. If the fatty acid chains are unsaturated, that means there's at least one carbon-to-carbon double bond and a resulting bend in the fatty acid chain. In this circumstance, the tails cannot be closely packed together and are less dense. Put 3 fatty acid chains with glycerol and you have the fat triglyceride, an important energy source. Attach long fatty acid chains to alcohol and you get waxes, lipids that play a role in water regulation of plants and animals. Perhaps the most common lipid, or common to AP Bio at least, is the phospholipid. Phospholipids are amphipathic with 2 hydrophobic fatty acid tails and a hydrophilic head. The hydrophilic head has a glycerol, a modified phosphate group, and often a choline group. The properties of phospholipids and their nature of forming a bilayer makes them the ideal molecule to make up all cellular membranes. Because the intracellular and extracellular environments are aqueous, membranes are bilayered with tails facing inwards. This allows certain cells and membrane-bound organelles to compartmentalize their functions. Additionally, different types of phospholipids in cell membranes allow the organism's flexibility to adapt to different environmental temperatures. Lastly, steroids, which have 4 fused carbon rings with attached functional groups. They look very different from any other lipid with hydrocarbon chains, but they'll still be nonpolar and hydrophobic. You can find steroids in cell membranes like cholesterol, as well as with signaling molecules like hormones. Genetic information is transmitted from one generation to the next through DNA or RNA. Both of these polymers are examples of nucleic acids, which are made of monomers called nucleotides. A nucleotide has 3 parts: a pentose sugar, a phosphate group, and a nitrogenous base, all covalently bonded together. Nucleic acids have a linear sequence with directionality, designated as 5' on the phosphate side of the sugar and 3' on the hydroxyl side of the sugar. It is important to recognize that double-stranded DNA is anti-parallel, meaning that one side of the backbone is oriented 5' to 3', while the strand across from it is 3' to 5'. Directionality will be very important when discussing molecular orientation and enzyme binding during processes such as replication, transcription, and translation. The difference between DNA and RNA sugars occurs at the 2' carbon. RNA has ribose, which contains a hydroxyl group at the 2' carbon. DNA has deoxyribose, which is missing the oxygen at the 2' carbon. A nucleotide's phosphate group is covalently bound to the 5' carbon of the pentose sugar within a nucleotide and to the 3' carbon of the pentose sugar of an adjacent nucleotide. Alternating sugars and phosphates form the backbone of each nucleic acid through the formation of a specific type of covalent bond called a phosphodiester bond. The phosphate group has a negative charge, which is why DNA samples migrate towards the cathode side during gel electrophoresis. Phosphate groups are the same in RNA and DNA. The last part of a nucleotide is the nitrogenous base, which encodes the genetic information. The nitrogenous base is arranged perpendicular to the backbone and covalently bonded to the pentose sugar at the 1' carbon. There are 5 different types of nitrogenous bases grouped into 2 categories. The purines are double-ringed nitrogenous bases and include guanine and adenine. These are found in both DNA and RNA molecules. The pyrimidines are single-ring nitrogenous bases and include cytosine, thymine, and uracil. Cytosine is common to both nucleic acids, whereas thymine is only in DNA and uracil is only in RNA. Adenine will always bond with thymine or uracil with 2 hydrogen bonds, and guanine will always bond with cytosine with 3 hydrogen bonds. To recap, water is an amazing molecule whose polarity influences hydrogen bonding and all of its properties, like cohesion, adhesion, and surface tension. Water is formed through dehydration reactions and added when dividing polymer chains during hydrolysis reactions. Organic molecules are made of carbon and form a variety of structures through bonding patterns with the elements hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Organisms obtain these elements from their environment and metabolize polymers to grow, reproduce, and maintain organization. The 4 major biological molecules are carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates are formed from monosaccharides and provide energy and structural support. Proteins are polypeptides made of amino acids which fold into specific shapes through hydrogen bonding and R group interactions. Lipids are nonpolar, hydrophobic molecules including fats, phospholipids, and steroids. And nucleic acids are the molecule of heredity with directionality formed from nucleotide monomers. Phew, we made it!